Hello, everybody. I welcome you all again to Unit 14 Biomolecules, Part 4. In the previous unit, we had learnt about carbohydrates. Now we shall discuss about proteins. What are proteins? Proteins are another important class of biomolecules which are essential for all living beings. Now, how did this name protein come? This is a Greek word, proteos, which means of prime importance. Proteins are very vital chemical substances which are essential for growth and maintenance of life. That is why the word proteose was used to name these class of compounds, biomolecules, and the terminology is therefore called proteins. Now, all the proteins are considered as the polymers of alpha amino acids. Now, what do you understand by the polymers of alpha amino acids is what is going to be studied in the next part of the video. But before that, we would like to definitely understand and try to analyze what are the sources from which you get proteins. So, if you are talking about the sources of proteins, then all the food products what we eat coming from plants and animals are the source of proteins. Now the question that comes to our mind is, where do you find proteins? Now it is found that proteins are present in almost all the living cells of plants and animals. In human beings, it is present in the hair, the skin, the nails, tendons, arteries, connective tissues, etc. Therefore, each living cell is made up of thousands of different proteins. So, therefore, proteins are all considered as the polymers of alpha amino acids. Amino acids. What is this term in telling us about? Amino acids, they are the building blocks of all peptides and proteins. Now, what do they contain? The amino acids contain the amino group NH2 group as well as the carboxyl group COOH functional group in the formula. And we are having the groups NH2 as well as the acid group attached to the carbon atom, which is called the alpha carbon atom. Now, this is the relative position of the amino group with respect to the principal functional group, carboxylic acid. So, therefore, the name is given here as alpha amino acid, as both the amino group and the carboxylic acid group is attached to the same carbon, the alpha carbon atom. Now, the group R, what does it indicate? This R is going to be all other different functional groups that could be present in the formula of the amino acid. The general formula, therefore, is summarized as RCHNH2COOH. The proteins generally on hydrolysis gives alpha amino acids. That is what we are going to get on the hydrolysis of the protein. Now, when we are having the study of proteins and thus amino acids, telling us that there are about 20 amino acids that have been identified as the constituents of proteins. The structures and their three-letter abbreviation and one-letter symbol, etc., are all indicated in this table here. Now, all the names of the amino acids, 20 amino acids, are all listed as the trivial names. Now, the trivial names generally reflect the property of the particular compound or the source from which the compound is obtained. Let's take one or two examples. Glycine. Glycine is very sweet to taste, and hence it has derived its name from the Greek word glycos, which means sweet. So that is why we are giving the name for this amino acid, which has got the R group to be a hydrogen, as glycine. So we have so many other amino acids listed here. Alanine, where the R group is a methyl, where valine, where the R group is an isopropyl group. Then you are having leucine with a long chain of carbon atoms, which is going to be the R group, and another long chain alkyl group, so on and so forth. Serine having hydroxyl group present. So the name of the organic compound and thus the amino acid is generally written on the basis of the source from which it has got or the property of the amino acid molecule which we are having with us. You can see one more example here, tyrosine. Now, the word tyrosine is obtained from source from where we get this amino acid. Tyrosine name is obtained from the the Greek word called tyros, that means cheese, and cheese is the major source from which we get tyrosine. So that is the way all these names of amino acids have been derived. This is some of the other list of amino acids which we are having. So altogether there are 20 amino acids which is found to be the constituent of protein. Now let us proceed towards the classification of amino acids. This is based on their nature. So based on the nature, the amino acids are classified as acidic, basic and neutral. So the simple terms of chemistry will help us to understand these amino acids better. If you talk about an acidic amino acid, here there will be more number of COOH groups in the compound when compared to the NH2 amino group, and hence Such amino acids are called acidic amino acid. For example, aspartic acid or glutamic acid, etc. Now, if you talk about the next category, basic amino acid, here the amino group should be more in number when compared to the carboxylic group, and hence these compounds are called basic amino acid. For example, lysine. Now when we go towards the neutral amino acid, it is noticed if there are equal number of amino and carboxylic acid groups in the formula, then it is called as neutral amino acid. Examples: glycine, alanine, serine, etc. So this is how you are going to classify the amino acids based on their nature. Let us now talk about the classification of amino acids based on the biological need. So we are having two broad classes. One, essential amino acids, and the other, non-essential amino acids. Now let us talk about non-essential amino acids. Here, non-essential amino acids, why do you call it like that? Let's try to understand. See students, there are 20 amino acids which are required for protein synthesis. The human body can synthesize only 10 amino acids, which are called, therefore, non-essential amino acids. So as our body can synthesize it, they need not be taken in the diet. So they are called non-essential amino acids. Example, glycine, serine, etc. But when we discuss about essential amino acids, so as I told you, we are having 20 different amino acids with us. Out of that, 10 are non-essential amino acids. The remaining 10 become essential amino acids. Now, the 10 essential amino acids are thus called because the human body cannot synthesize these amino acids, and therefore they are very essential to us. They are very much important for the growth of the living organism, and these essential amino acids therefore must be obtained through our diet, and deficiency of these essential amino acids will cause diseases like kwashiorkor. What is that? It is a disease where the water balance in the body is disturbed, and some organs because of that would become bloated. So this is one of the problems that could be caused if there are essential amino acids not taken through our diet. So the essential amino acids examples could be tryptophan, valine, isoleucine, etc. Now there must be obtained. How could you obtain these essential amino acids then? As I told you, it should be taken through our diet. So where a diet means what should be taken, consumed by us? It could be obtained from milk, meat, fish, eggs, beans, pulses. All of them provide the essential amino acids. Now, we need some method to remember the examples of essential amino acids and non-essential amino acids. So, one of the tools or techniques which we all used to adopt when we were students were using this term, mat will fly. That is the reason I've included this for your understanding and remembering of essential amino acids. Each word or each letter indicates one amino acid which is essential. So M stands for methionine, A stands for arginine, T stands for threonine, T stands for tryptophan, V valine, I isoleucine, L leucine, P phenylalanine, H histidine, Ly becomes lysine. So this is one of the ways to remember the examples for our essential amino acids. The rest of them are non-essential amino acids. Let us now understand the classification of amino acids based on the type of the R group that could be present. So they are classified into aliphatic, aromatic and heterocyclic amino acids. So it's very simple here. Aliphatic amino acid means there should be an open chain structure. Some examples are glycine, alanine, etc. Aromatic amino acid, the amino acids which contain essentially a benzene ring or an aromatic ring. Example, phenylalanine, tyrosine, etc. Heterocyclic amino acid, there should be an heterocyclic group in the structure, and example, histidine, proline, etc. When we talk about the properties of amino acids, it is noticed that the amino acids are usually colorless crystalline solids. Very interesting, is it not, students? We would think amino acids are liquids, but they are colorless crystalline solids. They are soluble in water and they have high melting points. Now, the most important aspect about the amino acid is that they behave like salts. Why should they behave like salt? They behave like salt due to the presence of both the functional groups, like the acidic carboxylic group COOH group, as well as the basic amino group NH2 group in the same molecule. So naturally, there could be a reaction between the acidic group COOH group as well as the basic amino group as they are present in the same molecule. So therefore, the reaction of an acid with a base will result in the formation of a salt, and hence amino acids behave like salts. This is one of the property of the amino acid which you must understand. The next property of the amino acid is the Zwitter ion. Spectroscopically, when experiment was done, it was found that the COOH carboxylic group and the NH2 amino group of amino acid do not exist as such, but they react from each other, and they react with rather each other, and hence we are observing that in the aqueous solution the carboxylic acid has the ability to lose a proton, because acids are proton donors and the amino group can readily accept a proton because bases are proton acceptors, and due to this we obtain a dipolar ion. This dipolar ion is called as Zwitter ion. You must be wondering, how is this Zwitter ion? It is a neutral structure. It contains both the positive and the negative charge. How? Let's look at the covalent form of amino acid. The amino group is the basic group ready to accept the proton. The carboxylic acid has got ability to donate a proton. So when it donates H plus ion from here, it is accepted by the basic NH2 group to become NH3 plus after accepting the proton. And C double bond O minus is the other part of the formula, as there are two opposite charges in the same molecule. The compound is a dipolar ion, a Zwitter ion. And now the amino acid molecules are found to be amphoteric in nature. Amphoteric means they can behave as an acid as well as a base. So in the Zwitter ion form of amino acid, the amphoteric behavior could be easily understood. The amino acids can react with acids as well as bases. So whenever we are talking about any amphoteric substance, it is a substance which is capable of reacting with both acid as well as with base, which is done by the zwitterion form of the amino acid. Hence, we are calling it as amphoteric in nature. Let us talk about the optical activity and configuration of amino acid. It is noticed that all amino acids are optically active except glycine. Now, why is amino acid optically active? It is active because it has got the alpha carbon atom, which is asymmetric. So therefore, they're all optically active compounds. Now, the molecule of rather glycine is the only one where there are two hydrogen atoms attached to the alpha carbon. Hence, it is not optically active. Now, the amino acids exist in the D and the L forms. So, it is observed most of the naturally occurring amino acids have L configuration. The L configuration has got the amino group on the left side of our carbon atom and hydrogen present on the right side. So this arrangement, this configuration is called as L amino acid, and the mirror image will be D amino acid. How the left hand's mirror image is the right hand, which is inverted, just like that. L amino acid's mirror image will also be like this, which is an inverted structure. But we find all the amino acids which occur in the nature to have which configuration? L configuration. Let us understand the structure of the proteins. Now, what are the structure of the proteins telling us about is the proteins are all condensation polymers of alpha amino acids. So, therefore, in the case of our proteins, there is a reaction between the carboxylic acid group and the amino group to have loss of water molecule called condensation reaction, and resulting in the formation of a peptide linkage, which is a CONH linkage between two amino acids. So what is a peptide linkage? Is generally a question asked in the exam. A peptide linkage or a peptide bond is an amide linkage. And what is an amide linkage? CONH linkage is called. the amide linkage, which is formed between two amino acids. What is then a dipeptide? A dipeptide is consisting of two amino acids where the acid group of one amino acid reacts with the basic group of another amino acid to give rise to a formation of a dipeptide. And here the peptide linkage is having a link between the CO and NH group of the amino acids. So therefore the peptide or a dipeptide will contain only one CONH linkage and it is having only one peptide bond. So let us understand this. When you are having glycine reacting with alanine, the OH group from the acid reacts with hydrogen of the amino group to be lost as water. As a result, the resulting structure has got CONH linkage, which is called as the peptide linkage. This is what is a dipeptide. It is formed by the combination or the reaction between two amino acid molecules. Let us now understand the different types of peptides. If you are having a dipeptide, you would have two amino acid residues, one peptide bond would be found. If there's a tripeptide, there would be three amino acid residues and two peptide bonds. If there is a polypeptide, there would be n number of amino acid residues and the number of peptide bonds would be n minus 1. This is the general formula used to find out the number of peptide bonds depending upon the number of amino acid molecules present. Now then, what do you understand by a polypeptide? If you are talking about a polypeptide, there are found to be the condensation polymers of large number of amino acids. So here the amino acids, if they are more than 10, then they are all called our polypeptides. Then what is protein? How do you differentiate between the polypeptide and a protein? A polypeptide with more than 100 amino acid residues with a molecular mass higher than 10,000 unified mass, U means unified mass, is called as the protein. So this is the basic difference between amino acid and protein. Classification of proteins. Proteins can be classified into two types on the basis of their molecular structure. They are fibrous proteins and globular proteins. These are the two types of proteins broadly classified. Now, when we are talking about fibrous proteins, the name itself tells us that it should be fibrous, linear, thread-like structures, and the polypeptide chains run parallelly and they're held together by hydrogen bonds, disulfide bridges, bonds, etc. The structure of protein is such that fibrous in nature and it is found to be insoluble in water because of those very strong bonding which is present, and those are the hydrogen bonds, etc., making it insoluble in water. For example, all the proteins which are structural material amino acids in the proteins, and which are forming the structure materials such as keratin, which is found to be present in the hair, wool, etc. Myosin, which is present in the muscles. Uh, then fibronin, which is present in silk. Collagen, which is present in tendons. All these come under the category of fibrous proteins. When we shift our attention towards globular proteins, here the structural arrangement such results in the formation of a globule or a spherical shape. And here there are the hydrophilic polar groups, which are all pointed outwards, and these interact with water and hence they are soluble. So therefore, globular proteins are found to be soluble in water. So examples: insulin, albumin, etc. Let us understand the basic differences between fibrous and globular protein. If you are discussing about fibrous protein, they are thread-like structure. Globular, they have spherical shape. Fibrous proteins are insoluble in water. Here, they are soluble in water. Polypeptide chains run parallel to each other. Here, they run in the form of coils, and fibrous proteins do not have any biological activity and they act as a structural material, whereas globular protein possess biological activity. So students, differences between fibrous and globular protein is quite a popular question. So when you are preparing, read this very well. So this is how the structure of a fibrous protein and a globular protein could be seen. The structure and the shape of the proteins. When we are going to understand the structure and the shape of the proteins, this is studied at 4 different levels. The levels are the primary, secondary, tertiary, and the quaternary structure. Each level is found to be more complex than the previous one. So this complex structure of the proteins can be understood as follows. The primary structure. Here in the primary structure, the sequence of amino acids in the particular polypeptide chain is understood. So here the different chemical and biological properties of the protein is only because of the primary structure. Students, you have to note this point that if there is any change in the primary structure, that means the sequence of amino acid, if it is changed in the primary structure, then the property of the protein is, biological activity of the protein is getting destroyed. Let us talk about the secondary structure of the protein. In the secondary structure, we are having the long polypeptide chains, how they exist, that is explained by this secondary structure. The secondary structure of the protein was studied in depth and proposed also by Linus Pauling. He proposed that there are 2 structural arrangements in the secondary structure for the protein, which are called the alpha helix and the beta pleated structure. The alpha helix which we are having is found to have the hydrogen bonding between the carboxylic group of the amino acid, which is going to form a bond with the amino group of the fourth amino acid residue in the chain. This is what you are having in the arrangement of the bonding between the C double bond O and NH group, giving rise to the helical structure. Some of the examples for the helical structure is the alpha keratin, which is found present in the hair. The beta pleated structure have the polypeptide chains which are arranged in a zigzag manner as the chains lie side by side. The chains are stacked one above the other, like how the pages of a book are arranged. This gives a three-dimensional structure for the protein. An example for the beta pleated structure is beta keratin, which is also present in the hair. So when we are talking about the tertiary structure of the tertiary structure of the protein always tells us about the overall folding of the polypeptide chains. And in this overall folding of the polypeptide chains, it gives rise to either the fibrous or the globular protein. The forces that stabilize the secondary and the tertiary structure of the proteins are hydrogen bonds, disulfide linkages, van der Waals forces, and electrostatic forces of attraction. So this is how the tertiary structure of the protein looks like. The quaternary structure of the protein, here we are having the proteins generally having two chains which are held together by hydrogen bonds, and the two or more polypeptide chains, which are referred to as subunits, are held with each other through different types of van der Waals interactions, hydrogen bonds, electrostatic forces of interaction, giving rise to extremely complex structure called as the quaternary structure of the protein. Denaturation of proteins is another one very important factor or a property of the protein which we must understand. This is very important from the examination point of view as well. The protein has a unique three-dimensional structure with a specific biological activity. Such a protein is called as native protein. When such a native protein is subjected to heat or changes in the pH or treatment with acids or alkali or some other denaturing agents, then this structure of the protein, the native structure of the protein, gets destroyed or disrupted. This behavior or this phenomenon is called as denaturation of proteins. Now what happens because of this? Because of denaturation of proteins, there is loss of biological activity of the protein, and therefore the protein will no longer be able to perform its biological function. What should be noted is during denaturation, the secondary and the tertiary structures are destroyed, but the primary structure of the protein will always remain intact. Some of the examples for denaturation of protein would be the coagulation of egg white, which on boiling will get converted into a mass, and that is how you can explain the denaturation of protein. The curdling of milk is another one example of denaturation of protein, which is caused by the bacteria which is present in the milk. The picture depicts this curdling of milk, explaining the denaturation of proteins. So students, with this we are coming towards the end of the study about proteins in this particular video, and the later part of the chapter will be continued in the next video which you are going to come across. So I hope all of you have taken down certain important points, right? We'll meet up with each other in the next video. Okay, bye bye. Thank you.